let's concentrate about antisense RNA. It is involved in gene regulation. It is involved in gene regulation. Sometimes it is possible for the DNA strand other than the DNA strand which sense RNA has been transcribed to be copied into RNA. The RNA produced by transcription of wrong strand of DNA is called antisense RNA. Its binding to sense RNA may physically block ribosome binding or elongation and thus may inhibit translation. Binding of antisense RNA to sense RNA may also trigger the degradation of sense RNA and thus block the gene expression. Now let's write here step by step. Antisense RNA helps in gene regulation. How? Sometimes it is possible for the DNA strand. All of we know that DNA strand have two strands as it is dsDNA. One is template strand and another strand coding strand. Though its name coding strand, but it does not code, not participate in coding. This coding strand not participate in coding, only template strand participate in coding. So sometimes it is possible for DNA strand. This strand normally this strand transcribed into mRNA, but other than this strand means from this coding strand this copied into RNA. Sometimes, not all time. All time the RNA, mRNA is transcribed from this template strand of DNA duplex. Sometimes this coding strand which is normally it is non-coded into RNA, but sometimes it can be coded into RNA. Sometimes, exceptionally. This one is not right strand is here. This one is wrong strand of DNA from which this RNA is synthesized or transcribed. This RNA is called antisense RNA. Is it very clear? So, antisense RNA is synthesized from the coding strand, a wrong strand of DNA duplex, DNA strand, not from template strand from which usually mRNA usually transcribed. This from this known wrong from this coding strand here from this wrong strand which RNA is synthesized, that one is known as antisense RNA and it binds to sense RNA, okay, and block ribosome binding or elongation and thus may inhibit obviously translation. And this binding of antisense RNA with this sense RNA, antisense RNA with this sense RNA may also trigger the degradation of sense RNA and thus block gene expression. So now let's summarize. It's about some special RNA, some type special RNA is antisense RNA. Antisense RNA helps in gene regulation. How antisense RNA is produced? Antisense RNA is produced from the wrong strand of DNA, not from template strand. Normally from the template strand mRNA is synthesized, they are transcribed, template strand transcribed into mRNA. From the wrong strand, which one is known as coding strand, but it does not code anyone normally. From this wrong strand of DNA, which RNA sometime, not always, sometime RNA is synthesized and that one is known as antisense RNA, not in sense, antisense RNA. When this antisense RNA go and binds to sense RNA, it can act two different ways. First of all, it can block ribosome binding on elongation and inhibit translation. Also, it can trigger, so when it binds with sense RNA, antisense RNA when binds with sense RNA, it can trigger degradation of sense RNA and block gene expression. Either it can inhibit translation, either it can block gene expression. Okay. Either when it block ribosomes binding or elongation, then it can inhibit translation, and when it trigger the degradation of sense RNA while after binding sense RNA, it blocks gene expression. So it's all about antisense RNA. Now come after discussing all of this. Now I am going to discuss about the last type of this special RNA. That one is LNC RNA. Is long non-coding RNA. It helps in long non-coding RNA chromatin modification. It helps in regulation of transcription, splicing, miRNA and siRNA. And the third, this one is 200 nucleotides long. So it's all about, let's summarize the long non-coding RNA. Obviously it is the non-coding as the name suggests. This one is 200 nucleotide long and it helps in chromatin modification and it helps regulation of transcription, splicing, miRNA and siRNA. So it's all about some special type of RNA.